All right, welcome everyone. Today we're jumping into one of the most incredible stories in all of biology. We're talking about the transformation from a single cell into the fundamental blueprint for a human being. And all of this happens in just the first 3 weeks. It's an absolutely epic journey playing out on a stage so small you can't even see it.
So our story really kicks off with this number right here, 200 million. That's about how many sperms start this incredible journey, all racing towards a single oocyte. This isn't just a process, It's a competition on a scale that's almost impossible to imagine.
I mean, to put that into perspective, this is more than a race. It's like a lottery with just insane odds. Out of this massive crowd, only the absolute best, the strongest, the most capable, are even going to get close to that finish line.
But before a race like that can even begin, the contenders, the messengers, they have to be perfectly engineered for the job. So this first part is all about how these incredibly specialized cells are actually made.
And right away, you see this huge difference in evolutionary strategy. For males, spermatogenesis is all about quantity. You get one primary cell, and it turns into 4 lean, fast, motile sperm. The whole idea is to maximize the chances that at least one of them makes it. But for females, oogenesis is all about quality. It starts way back, before birth, and one cell pours all of its resources into making one single, massive oocyte that's packed with all the nutrients needed to kickstart life.
So let's zoom in on that final transformation for the sperm, a process called spermiogenesis. This is where a regular round cell becomes a tiny, biological high-performance machine. First, the acrosome forms. Think of it like an enzymatic warhead on a missile. Then, the nucleus gets super compact, packing all that genetic data in tight. The neck and tail form, which is basically the engine. And finally, and this is key, it sheds all of its extra cytoplasm. It ditches every last bit of unnecessary weight to become perfectly streamlined for one thing: speed.
Okay, so our contenders are built, they're streamlined, they're ready to go. Now, it's time for the race itself. And this is no simple swim. It's a brutal obstacle course filled with biological traps and challenges that, honestly, only the absolute best can overcome.
So what's the first huge challenge? Getting past the oocyte's incredible defenses. It is literally surrounded by protective layers. It's a fortress, and to breach it, you need a very specific set of keys and tools.
This is where something called capacitation comes in. And you can totally think of this as the sperm's essential 7-hour-long training montage. During this conditioning period, a protective coat is stripped away from the sperm's head and its tail kicks into hyperdrive. This isn't just a warm-up, it's the critical arming sequence that makes fertilization even possible.
So, once a fully armed and capacitated sperm finally reaches the oocyte's outer shell, the zona pellicuda, it binds to a special protein, ZP3. That binding is the trigger. It sets off the acrosome reaction. The cap we saw being built bursts open, releasing its enzymatic toolkit. Enzymes like acrosin get to work, literally digesting a path right through that final protective barrier.
But wait a second. The moment one sperm makes it through, a brand new, critical problem pops up. What if 2 or 3 or even more get inside? That would be lethal. So the oocyte has to have a way to instantly slam the door shut behind that first winner.
And its 2-step security system is just brilliant. The instant the first sperm fuses, you get the fast block. The oocyte's membrane depolarizes in a flash, creating a temporary electrical force field that just repels any other sperm. Right after that comes the slow block. Enzymes are released that permanently change the outer shell, destroying all the receptor sites. It's like slamming the door shut and then immediately changing all the locks.
So, fertilization is complete. We now have a zygote, but its journey is nowhere near over. This brand new cell now has to travel to a safe place and set up a home for the next 9 months.
So here's a quick look at that first week's travel log. Day 1. Fertilization happens way up in the uterine tube. Then, while it's traveling, it's dividing over and over again. Around day 5, it's now called a blastocyst, and it actually hatches out of that protective shell. And by day 6, it starts the super important process of implantation, burrowing itself into the rich, welcoming wall of the uterus.
And when it comes to implantation, location is everything. The perfect, normal spot is high up on the uterine wall. But, sometimes it doesn't work out that way. If the blastocyst implants somewhere else, it's called an ectopic pregnancy. And get this, over 95% of those happen in the uterine tube, which is a life-threatening medical emergency. It just goes to show how critical this final step of finding the right home really is.
Okay,
So once it's successfully implanted, the pace just explodes. In the second and third weeks, that simple little ball of cells starts this incredibly rapid and complex process of organizing itself, basically laying down the fundamental blueprint for the entire body.
The second week has this really cool nickname, the Week of Twos. And for good reason. It's this beautiful, symmetrical period where 4 key structures each split into 2. First, the trophoblast. That's the outer layer handling implantation. It divides. Second, the embryoblast. The inner cells that will become the embryo divides into 2 new layers, the epiblast and the hypoblast. Third, 2 new cavities pop up, the amniotic cavity and the yolk sac. And finally, a support layer, the extraembryonic mesoderm, also splits in 2. It's this perfectly organized doubling that sets the stage for everything that's about to happen.
Now, just take a breath for a second. Because of all those layers we just talked about, it turns out that all 3 of the primary germ layers, the very foundations for every single tissue and organ in the body, they all come from just one of them, the epiblast. It is an absolutely astounding, mind-blowing feat of biology.
And this incredible architectural process is called gastrulation. It's the main event of week 3. Cells from the epiblast start moving toward a groove called the primitive streak, and they just dive inward. The very first wave of cells to dive in pushes the old hypoblast out of the way to form the endoderm. The next wave of cells fills in the middle space, creating the mesoderm. And the epiblast cells that just stayed on the surface, they become the ectoderm. So, in just a matter of days, a simple 2-layer disc is completely rebuilt into the 3-layered foundation of a human being.
So there you have it, from a race between 200 million to the intricate architectural dance of gastrulation, a complex blueprint for life gets established in just 21 days. It's a process of just stunning precision, efficiency, and profound change, which I think leaves us with one last thing to think about. What does this microscopic journey really, truly reveal about the nature of life itself?